Imagine you're in the produce aisle of an unfamiliar grocery store and looking for the zucchini. Although you're currently looking at the mushrooms, within a second or two you make a saccadic eye movement that lands on the target of your search. One way to explain how you did that is in terms of computational processes operating on cognitive representations. A different explanation would be in terms of neurophysiology, the cascade of neural spiking activity that culminates in the production of a saccade. Although these explanations seem distinct, I'll briefly describe a model called SCRI that bridges the gap between these levels of description. SCRI stands for Salience by Competitive and Recurrent Interactions. SCRI was developed to understand why neurons in frontal eye fields, FEF, act the way they do during visual search. Single-unit spiking activity of individual FEF neurons was recorded from rhesus macaques during visual search. This example illustrates a typical setup in which the monkey is rewarded for making a saccade from a central fixation point to a designated target object, here the T shape, embedded within a display of L-shaped distractors. FEF is critical for visual search because it is the confluence of two processing streams, a dorsal stream that carries localization information and a ventral stream that carries identification information. We focus on two types of neurons in FEF. One type, which we call movement neurons, enacts an accumulation process whereby the neurons ramp up spiking activity to a threshold. The neurons that first reach threshold determine both the timing and endpoint of a saccade. The other type, visual neurons, show a characteristic three-phase pattern. After an initial period of low baseline activity, visual neurons begin to ramp up their spiking in response to anything in their receptive field, whether target or distractor. If the object in the receptive field is a target, activity keeps increasing and stays high. If it is a distractor, its activity starts to diminish. SCRI explains these neural dynamics in terms of how FEF visual neurons integrate localization and identification signals and interact with one another. When an object appears in the receptive field of an FEF visual neuron, areas like MT send a pulse-like localization signal indicating that something is there. This causes the FEF neuron to spike more, whether the object is a target or distractor. This increased FEF activity acts as a recurrent multiplicative gate, telling areas like V4 and temporal areas to compare the features at that location with those of the search target. This identification signal, in turn, excites those same FEF visual neurons to the extent that the features at that location match those of the search target. Thus, neurons with a target in their RF will tend to maintain a high level of spiking activity, while those with a distractor will no longer receive as much excitation. The localization signal opens the door, but identification information can keep it propped open. In addition to these excitatory connections, feed-forward and lateral inhibitory connections can act to amplify differences in activity between neurons to help resolve the competition between potential saccade endpoints. By exciting movement neurons, FEF visual neurons act to construct a dynamic representation of salience evidence that is then accumulated by FEF movement neurons to drive the production of a saccade toward a selected target. SCRI predicts the millisecond by millisecond spike rate of individual and often idiosyncratic FEF visual neurons, of which 6 are shown here. Across all 94 neurons in our dataset, SCRI accounted for an average of 87% of the variance in spiking activity that FEF visual neurons produced during visual search. This includes accounting for how those spiking dynamics are modulated by increasing the number of distractors, set size, or by increasing the similarity between distractors and the search target. Because FEF visual neurons drive the movement neurons that produce saccades, SCRI also explains in quantitative detail how set size and similarity affect saccade behavior. Here, the points are quantiles of the saccade response time distributions produced by different monkeys in this task. Smooth lines are the saccade timing distributions produced by the model. In summary, SCRI accounts for the momentary spiking activity of individual neurons as well as the whole organism saccade behavior driven by those neural dynamics. SCRI can bridge these scales because SCRI bridges computational and neural levels of description. This bridge goes both ways. Neurophysiology provided the empirical constraints necessary to characterize the localization and identification signals, as well as the types of possible interactions present in SCRI, which enable it to account for whole organism behavior. On the other hand, by explaining neural spiking in computational terms, we can understand the diversity of FEF visual neurons in terms of the functional roles those neurons play in producing whole organism behavior. Although I haven't been able to get into it here, this second point leads to specific testable predictions about neurophysiology, and I encourage interested viewers to check out our paper for more details on this and many other issues. More broadly, I believe SCRI illustrates the power of the burgeoning field of model-based cognitive neuroscience to unite researchers from different areas and lead to a more comprehensive understanding of mind and brain that reaches across levels of description. With that, I would like to thank my fantastic collaborators that made all this work possible, and all of you for your time and attention.